Question 1, genes are, we have four options, DNA, RNA and proteins. B option states specific segments of DNA, C with DNA plus related RNA, and D option with DNA and histones. First of all, DNA and RNA both which we have are nucleic acids, and proteins is a sequence of amino acids. Genes are a specific segments of DNA, first of all, which they have directly stated in that option. So we need to understand about genes first. Genes are just these specific segments of DNA which codes for a specific trait and which get expressed. Okay, so that's what the direct answer, that B with specific segments of DNA. Second question, the genetic material in viruses is DNA, first option, DNA or RNA, C option RNA, and D option DNA and RNA. First of all, viruses are on the border of living and non-living, that we need to understand. So in the host they become active, outside the host they are inactive, which we call them as non-living, since they do not replicate on their own. The B and D option states or and. It has both the nucleic acid as a genetic material, but not at the same time. They have either DNA or RNA as their genetic material, not both. So A option is ruled out, C and D is also ruled out. B is the correct answer. Third question, Griffith effect is about what? A option tells us DNA transcription, B DNA RNA translation, C bacterial transformation, and D bacterial transduction. Frederick Griffith was a scientist who took two strains of bacteria known as pneumococcus. One was smooth strain, another was rough strain. He mixed both. So smooth strain was heat killed since it was virulent, and rough strain was non-virulent. So he injected them into mice, which resulted into death of the mice. So there was a certain principle which got transformed, transformed from the virulent strain to the non-virulent strain. This transforming principle was later found out to be DNA. So since it was a transformation of one type of bacteria into another, it is known as bacterial transformation. So C is the correct answer. Question number 4, in Streptococcus pneumoniae, A option tells us that is it virulent form is smooth, B virulent form is rough, C non-virulent form is capsulated, D all forms are rough. Streptococcus pneumoniae is a bacteria which was used in experiment by Griffith, scientist. He used two strains, the smooth and rough. Why smooth and rough? Because on culture medium they produced smooth colonies and other strain produced rough colonies. So smooth ones have capsules, which is their outermost covering. Rough ones, they do not have capsule, no capsule, so which do not offer protection against phagocytosis by macrophages. They do not offer protection, so that is why the smooth ones which have capsule offers protection against phagocytosis by macrophages. That is why virulent form is smooth, is the correct answer. Question number 5, the transforming principle of pneumococcus as found out by Avery, MacLeod and McCarthy was option A mRNA, option B DNA, option C protein, and option D polysaccharide. Again the same story of Griffith. He used two strains again, smooth and rough. Smooth transferred his transforming principle when they were mixed. This transforming principle was later found out by Avery, MacLeod, and McCarthy to be DNA. So this B, option B is the correct answer. Question number 6. What happened when it killed S cells along with live R cells were injected into mice? Option A states mice died and showed live S cells. B, mice survived and showed live S cells. And C, option states mice died and showed live R cells. And D, mice died and showed dead R cells. Again, the Griffith experiment used S strains which were hit killed, which were mixed with R strains which were living. So, which when injected into mice, the mice died because there was a transforming principle which was transferred from S strain to R strain, which made it get converted into S cells, so which were living into the mice. So option A is the correct answer, that mice died and showed live S cells. Question number 7. Smallest gene affected by mutation is option A muton, option B cistron, option C recon, and option D exon. First we need to understand all these four options are types of genes. Types of genes in which muton is for mutation, cistron is a functional, recon is for recombination, and exon is the expressive gene. So they have asked the question, smallest gene affected by mutation is option A is the correct answer, since it is responsible for mutation, muton. Question number 8. The connecting link between generation is option A cell, option B nucleus, option C nucleic acids, and option D cytoplasm. Connecting link between generation means the one which connects both the generation. There is first as well as the second generation. So from the first generation to the second generation, there is only one thing which is transferred from parents to their offsprings, which is their genetic material. And this genetic material is known as nucleic acids. In eukaryotes, it is DNA, and in prokaryotes, it may be DNA or RNA. So the option C is the correct answer. Nucleic acids are the connecting link between generation. Question number 9. Find out the true statement. Option A, DNA is present only in the nucleus. No, DNA present outside the nucleus, which we call as plasmid. Option B, DNA is always linear. No, it is helical as well. C, DNA is synthesized by RNA. Not always. D, DNA can be self-replicated is important, as DNA has its one property of semi-conservative self-replication. Where the daughter DNA strands are synthesized from parent strands. So option D is the correct answer. DNA can be self-replicated. Question number 10. Nucleotides are made up of option A sugar and nitrogen base, option B nitrogen base and phosphate, option C sugar and phosphate, option D sugar, nitrogen base and phosphate. In this question you need to understand two terms, that is nucleotides and nucleosides. Nucleotides consist of a sugar linked to a nitrogen base which is linked to phosphate group, whereas nucleoside consists of nucleotide minus phosphate group. It does not have phosphate, nucleosides, whereas nucleotides has phosphate. So this is the basic composition of a nucleotide and this of nucleosides. So the correct option is option D. It is made up of sugar, nitrogen base and phosphate. Question number 11 states that DNA is a mature molecule because option A, it replicates. Yes, it has a property of semi-conservative replication. Option B, it is a genetic material. True. Option C, it controls metabolic activities by a process of transcription and then translation. So all three options are correct and option D states that all the above. So option D is the correct answer, that it replicates, it is a genetic material and it controls metabolic activities. So all the options are correct. So option D, all of the above are the correct answers. Question number 12, which states which of the following is correct pair of pyrimidine base. Option A, adenine and thymine, AT. Option B, adenine and guanine, AG. And option C, thymine and cytosine, T and C. And option D, guanine and cytosine. For this question to understand, you should know that there are two nitrogen bases, there is purines and pyrimidines. Purines are adenine and guanine and pyrimidines are thymine and cytosine. So they have asked the question which of the following is correct pair of pyrimidine base. So thymine and cytosine is the correct pair. So option C is the correct answer. Question number 13. The nitrogen bases are linked to sugar by. Nitrogen base linked to sugar by option A, phosphate bonds, option B, hydrogen bonds, option C, glycosidic bonds, and option D, phosphodiesterase bond. For this question to understand, you should know the basic structure of DNA. It consists of a pentose sugar which is linked to nitrogen base by glycosidic linkage, and it is linked to the phosphate group by phosphodiester bond. And these two nitrogen bases are linked to each other by hydrogen bonds. So option B is hydrogen bonds, option D is phosphodiesterase, and option C is glycosidic linkage. So the correct option for this question, which states that nitrogen base which is linked to sugar by glycosidic linkage. So C is the correct answer. Question number 14. which of the following is incorrect? Option A, adenine and thymine. Option B, guanine and cytosine. Option C, thymine and adenine. And option D, adenine and guanine. For this, you should understand the concept that adenine and guanine are purine bases. Thymine and cytosine are pyrimidine bases. Adenine is linked to thymine by 2 hydrogen bonds, and guanine is linked to cytosine by 3 hydrogen bonds. So it can be like this as well, that thymine is bonded to adenine by 2 hydrogen bonds and cytosine by 3 hydrogen bonds to guanine. So all these sequences are correct from this question. Option A is correct, option B as well as C is incorrect. So incorrect one is option D. So D is the correct answer. Question number 15, which states which is true according to Chargaff's rule. Option A plus G is equal to T plus C. Option B, A is equal to C. And option C, guanine is equal to thymine. And option D, adenine plus thymine to cytosine plus guanine is equal to 1. So for this question to understand, you should know Chargaff's rule, which states that purines to pyrimidines ratio is always equal to 1. That is, purines, which is adenine and guanine, are always equal to thymine and cytosine. Adenine and guanine are always equal to thymine and cytosine. That is, purines are equal to pyrimidines. So option A is the correct answer, that adenine plus guanine is equal to thymine plus cytosine. Question number 16, Okazaki fragments are option A, RNA primers. Option B, short DNA fragments on leading strand. Option C, short DNA fragments on lagging strand. And option D, DNA fragments from radiation action. For this question, you should understand the DNA replication process, which consists of 2 strands, that is 5' to 3' and from 3' to 5' polarity. On one strand, the replication is continuous. On the other strand, it is discontinuous. So this, we call it as leading strand, since it is continuous process of synthesis, and this as lagging strand, since this is discontinuous synthesis. These small fragments which have been synthesized of DNA are nothing but Okazaki fragments. Okay, so these are the Okazaki fragments, are small fragments of DNA which have been synthesized on discontinuously on lagging strand. So the correct option is option C, which states that short DNA fragments on lagging strand. Question number 17, stating DNA is acidic due to option A, sugar, option B, purines, option C, phosphoric acid, and option D, pyrimidines. First, let us rule out the incorrect options. Purines and pyrimidines are nitrogen bases. They are basic in nature. Sugar is also ruled out. Remaining is phosphoric acid, which gives it the acidic nature, which gives DNA acidic nature. So option C is the correct answer. Question number 18, Okazaki fragments are formed on option A, leading strand, option B, both these strands, option C, lagging strands, and option D, none of these. In the semi-conservative replication process of DNA, which occurs in 2 types, that is continuous and discontinuous part. So the continuous one we call it as leading strand, and the discontinuous one as lagging strand. This lagging strand shows discontinuous synthesis with short DNA fragments, which we call it as Okazaki fragments. So Okazaki fragments are formed on lagging strand. So option C is the correct answer. Question number 20. The replication of DNA is helped by option A, DNA polymerase only, DNA ligase, option B, DNA ligase only, option C, DNA polymerase and DNA ligase, and option D, none of this. Again, the replication part of DNA, semi-conservative replication, two strands leading and lagging. So this, on leading strand there is continuous synthesis, on lagging strand there is discontinuous synthesis, and there is discontinuous synthesis. These small fragments of DNA, which we call it as Okazaki fragments, they are been joined to each other by enzyme known as DNA ligase, and the enzyme responsible for continuous polymerization or synthesis of DNA is DNA polymerase. So for continuous synthesis we require DNA polymerase, and for discontinuous as well as with DNA polymerase we require DNA ligase as well. So to complete the semi-conservative replication of DNA we require both DNA polymerase as well as DNA ligase. So option C, DNA polymerase and DNA ligase is the correct answer. Question number 21. The components of nucleosome are option A, histones, option B, amino acid, option C, DNA, and option D, both 1 and 2. First, you should understand what is nucleosome. Nucleosomes are nothing but acidic DNA which is wrapped around octamers of histones. Octamers of histone. So this is a DNA and these are histones, proteins. So this DNA is acidic in nature and histones are basic in nature, due to which they get bind strongly. So nucleosomes are the structures consisting of acidic DNA binded to octamers of histones, which are basic in nature. So the components of nucleosome are both histones as well as DNA. So option D with both A and C is the correct answer. A and C is the correct answer. Question number 22. The central core of nucleosome contains how many molecules of histone? Option A 4, option B 16, C 8, and option D 32. Again, nucleosome is an octamer of histones around which the DNA is wrapped. So there are 8 molecules of histones which are been wrapped around that. So option C is the correct answer. Question number 23. Find the odd one out. Option A UAA, option B UGG, option C UAG, and option D UGA. First we need to understand what are these options. They are just codons, triplet codons, which codes whether to initiate or stop the synthesis, the protein synthesis part. Out of these 4 options, UAA, UAG, and UGA, these are stop codons, which terminates the process of protein synthesis. So the odd one out is option B with UGG. So correct answer is option B, UGG. Question number 24. Which of the following sequence is not translated completely? Option A AUG, GUA, CAG, and UAC. Option B AUC, UCC, UUA, UGG. Option C AUG, triple U, CAG, and UAA. And option D GUG, GCA, and EGG, and USC. We know that there are stop codons, which are UAA, UAG and UGA. In this options there is a stop codon out here, so this sequence will not be translated completely. So option C is the correct answer. Question number 25. Which of the following is central theme of protein synthesis? First of all, you should know that central dogma or theme of protein synthesis was put forth by scientist named Francis Crick, which was DNA undergoes own replication, which gets undergoes transcription to protein mRNA, and this mRNA undergoes translation to form proteins. So this is the central theme or dogma of Francis Crick. So out of these options, option A states DNA undergoes transcription from mRNA, mRNA translated into protein, transcript to mRNA, and translation into proteins, and option C is this. So option A is the correct answer, which states that DNA undergoes transcription to form mRNA, mRNA then undergoes translation to form proteins. So option A is the correct answer. Question number 26. Reverse transcriptase. A. Disintegrates host DNA. B. Polymerizes host DNA. C. Translates host DNA. D. Transcribes viral RNA to cDNA. You should know that reverse transcriptase is the enzyme which is present in HIV virus, which causes transcription of RNA into DNA. So option D is the correct answer. Question number 27. DNA undergoes C to form mRNA, which undergoes B to form protein, and which was proposed by, this theme was proposed by. So in this figure gives an important concept in the genetic replication of DNA or implication of DNA. Fill in the blanks A, B and C. So this is a central dogma which was proposed by Francis Crick, which states that DNA undergoes transcription to form mRNA, it undergoes translation to form proteins. So C is transcription and B is translation, and A option is Francis Crick. So option B is the correct answer, which is Francis Crick, translation and transcription. Question number 28. Which one of the following codons codes for the same information as UGC? A. UGU, B. UGA, C. UAG and D. UGG. Cytosine complements in DNA with thymine. In RNA, it complements with cytosine with uracil. So here the correct option is option A, since it is UGU with same complement as C. Option A is the correct answer. Question number 29. Enzyme needed for production of DNA from RNA is RNA polymerase? No. RNA polymerase uses RNA to form RNA. Option B, reverse transcriptase. Option C, DNA helicase. Option D, DNA polymerase. DNA polymerase DNA to form DNA. Reverse transcriptase is the enzyme which is used by HIV virus, which causes reverse transcription. This reverse transcription is nothing but RNA forms DNA with help of these enzymes. So correct option is option B, reverse transcriptase. Question number 13. In Escherichia coli, lac operon is induced by A. Lactose, B. Promoter gene, C. Beta galactosidase, and D. I gene. First of all, lac operon is nothing but polycistronic gene, where the repressor binds to the operator part to inhibit the synthesis of structural proteins by Z, Y, and A genes. But lactose, it binds to this repressor and thus prevents the inhibition of the operator gene, and which results into the synthesis of structural proteins. So thus lactose acts as an inducer for the lac operon. So the correct option is option A, lactose.